Hi, my name is Lauren Sims, and this is my final presentation for microbiology. So the first question that was asked was what our favorite topic was over the past session, and my favorite was microbial infections and immunology and epidemiology. Microbial infections was one of the most interesting topics to learn about because it taught me so much about different microbes and how they affect the body differently. I've known about or even personally experienced some of the microbial infections that we discussed during the lecture, so it was interesting to learn more about them and to learn about new microbial infections as well. Immunology and epidemiology was also really interesting to me. As a soon-to-be nurse, I find the human body fascinating, so to learn all the ways that the body takes care of itself automatically was very cool to learn about. Epidemiology was also super interesting given the recent pandemic and learning how the world prepares to combat different diseases. Again, as a future nurse and the possibility of being on the front lines in the case of another pandemic or just diseases in general, it was interesting to learn about how important this field of study is and what they focus on and what they do on a day-to-day basis. So for the second question, in the case that I was exposed to an unknown microbe, I would have to conduct a series of tests to figure out what it was and how to treat it. So some of the tests that could be conducted to see what microbe it is would be using microscopes to observe the structure of the cells, biochemical tests, serological tests, and protein tests for a more rapid identification. And to see if the microbe was a fungi, I would look for the cell wall containing chitin and membranes with egosterol. If it was a virus, I would look for a cell with nucleic acid surrounded by a capsid or an envelope. So just more about what the structure of that cell particularly looked like compared to the others. And I would also look for the shape of the cell to see if it was helical or complex, which are distinct from other cells. So really just focusing on what the structure was in different components. For bacterial cells, I would look for different structures with cell walls, cytoplasm, ribosomes, and what their DNA looks like. They also have the possibility of having a flagella where the other two typically do not. Bacterial cells also contain peptidoglycan, but no introns or a membrane-bound nucleus. So basically just the structures of the cells would be different. So depending on what all of that would look like would determine which kind of microbe it was, and then further testing such as gram staining to figure out exactly what type we're dealing with. So the third question is over vaccines, and if I were to encounter an individual who was against the use of vaccines or didn't fully understand the success in them, I would use my knowledge from this course to convince them otherwise, and I'd first explain the body's natural defense systems to invaders, such as viruses and bacteria. So the body has two types of protection for itself: innate and adaptive immunity. Innate immunity being routine protection of the body that's passed down from generation to generation through either vaginal birth and breastfeeding, from mother to infant. So this type of immunity is basic protection from day-to-day invaders and is why we have mucous membranes, inflammation, fevers, just kind of your body's first way of dealing with any unknown pathogens. Additionally, there's adaptive immunity, which requires an invader that is unknown to the body to come in and make us sick before the body can really learn how to combat it. After the disease or sickness runs its course, the body is able to create memory B cells that will remember this pathogen and destroy it next time it invades the body to prevent us from getting sick again. So, unfortunately, there are multiple viruses and bacteria and other microbes that can cause severe harm to the body and even death if they're not eliminated right away. And some microbes even exist that we don't have treatments for. So with the use of vaccines, we're able to introduce the body to a very small and practically harmless amount of the pathogen that will let the body learn how to attack the pathogen the same way it would if we were actually infected with the microbe in the first place. So the use of vaccines will allow us to have those memory B cells and a stronger immune system without having to weaken it at all by being sick first. The way that I think of it is that it's almost a way to cheat the system and kind of have kill codes before the problem even arises. So your body's basically able to have all the knowledge of how to destroy these pathogens without having to even deal with being sick in the first place. So for the fourth question is about microbial relationships and humans. So some relationships could be mutualistic, where both entities benefit from the presence of the other, or some may be parasitic, where one benefits and the other one is harmed. So some beneficial roles of microbes in the human body would be the various lactobacillus bacteria found in the vaginal canal of women. These microbes produce lactic acid and hydrogen peroxide that help prevent the growth of yeast and other unwanted organisms that just keep the female body healthy. Humans also use microbes to make food and alcohol. For example, humans use lactic acid producing bacteria in the production of yogurt. We learned about this in our recent lab of making yogurt, which was super interesting. This bacteria produces lactic acid that lowers the pH and causes the protein in milk to coagulate, forming thick yogurt that we then consume. Unfortunately, not all microbes have the most beneficial relationship with humans. Multiple microbes exist that can cause a lot of infection and harm to the body if they do get inside. So the Streptococcus pyogenes bacteria is what causes the common strep throat. This bacteria is highly contagious and spreads through droplets in the air. This bacteria takes root in your throat, causing pain, inflammation, fevers, and white patches in the throat. If you've ever had strep throat, we all know it's horrible. So that's just one of the pathogens or microbes that don't get along well with the human body. Another microbial infection is malaria. So this shows a parasitic relationship as the infection is a protist that uses the host for nutrients while the human body suffers. So being infected with malaria, you can take different medications to clear it, but your body definitely suffers while the protist is in your body. And for the last question is the different uses of microbes. So one of the many uses of microbes is in food production. I covered this a second ago in the yogurt context, but another way that it works is in alcohol fermentation. So yeast will ferment simple sugars and produce ethanol and carbon dioxide, and so that will then create the alcohol that we can consume. Microbes also contribute to the carbon cycle by decomposing organic molecules. They also help in the digestive tract of herbivores because microbes are able to digest cellulose, which is a major part in plant material, so it's easier to digest for the animal to then use as nutrients. Another way microbes can help is in water treatment. The lecture over this, we learned of a lot of different ways and how the entire process works. The specific point that I'm going to make is how aerobic microbes assist in water treatment in the secondary process by oxidizing any biologically degradable material to carbon dioxide and water. So that is able to treat the water and move it along in the process so we can have healthy drinking water and safer for the environment. The last way microbes can help is in composting. So composting works by having microbes decompose organic material into a form humans can use in gardens and for farming. So composting definitely giving us fertilizer and making healthier soil for us to use to grow our own food. So those are the 5 questions for the final presentation, and overall I really enjoyed this session and learning all about the different areas of microbiology. Thank you.